Burkitt lymphoma is a particular type of cancer that affects part of our body's defense system, specifically the B cells. These B cells are a kind of white blood cell, and their job is to help us fight off infections by making special proteins called antibodies. When someone has Burkitt lymphoma, these B cells start to grow and multiply much too quickly, forming tumors. It is known as one of the fastest growing human tumors, meaning it can develop and spread very rapidly if not treated promptly. This rapid growth makes it a very serious condition, especially for children who are often affected. Understanding what it is and who it affects is the first step in tackling it. This type of cancer is not spread evenly around the world. It is seen much more often in certain specific regions. It is particularly common in areas of equatorial Africa, such as Kenya, Uganda, and Nigeria, and also in Papua New Guinea. In these places, it is often referred to as endemic Burkitt lymphoma because it is consistently present within those populations. The rates can be significantly higher here, sometimes as much as 10 times more frequent compared to other parts of the globe like Europe or North America. This geographical pattern has puzzled scientists for a very long time, leading them to search for environmental factors or other diseases common in these areas that might be playing a crucial role in its development. The most common form of Burkitt lymphoma, the endemic type found in these specific regions, predominantly affects children. Young boys seem to be at a slightly higher risk than young girls. The symptoms can be quite distressing and noticeable. Often the first sign is a large swelling, most commonly in the jaw or other facial bones, which can grow very quickly. Sometimes the cancer can affect organs in the abdomen, leading to swelling of the belly, pain, and other digestive problems. Because it grows so fast, seeking medical attention as soon as these symptoms appear is incredibly important for giving children the best chance of successful treatment and recovery from this aggressive illness. For many decades, researchers and doctors working in these high-incidence areas observed a fascinating and worrying pattern. They noticed that Burkitt lymphoma seemed to occur more frequently in places where another serious disease was also very common. This observation, dating back to the late 1950s, was a critical clue. It suggested that there might be a hidden connection, a link between this other prevalent illness and the development of Burkitt lymphoma in children. This other illness, as we now understand much more clearly, is malaria, a disease spread by mosquitoes. The mystery was why and how these two very different diseases could be so closely intertwined, a question that recent science has started to answer. Malaria is a life-threatening disease that is caused by tiny parasites. These parasites are from the Plasmodium family. People get malaria when they are bitten by an infected female Anopheles mosquito. When the mosquito bites, it injects the parasites into the person's bloodstream. Once inside the body, these parasites travel to the liver where they mature and multiply. Then they infect red blood cells, causing them to burst, which leads to the common symptoms of malaria like fever, chills, sweating, headache, and muscle pains. Malaria is a huge public health problem in many tropical and subtropical parts of the world, affecting millions of people each year and causing many deaths, especially in young children and pregnant women, if not treated quickly and effectively. The link between Burkitt lymphoma and malaria is specifically tied to one particular type of malaria parasite, Plasmodium falciparum. There are several types of Plasmodium parasites that can infect humans and cause malaria, but Plasmodium falciparum is the most dangerous and the most prevalent in Africa. It is this specific species that has been strongly associated with the higher rates of Burkitt lymphoma. This is an important detail because it suggests that there is something unique about how Plasmodium falciparum interacts with the human body, especially a child's developing immune system, that creates a pathway towards this particular cancer. Other types of malaria, while still serious, do not show this same strong connection to Burkitt lymphoma. The connection between Plasmodium falciparum malaria and Burkitt lymphoma has been recognized by scientists and doctors for a very long time, stretching all the way back to the late 1950s. Epidemiologists who study disease patterns noticed that the geographical areas where Burkitt lymphoma was common were almost exactly the same areas where Plasmodium falciparum malaria was rampant. Children living in these malaria belts were not only at high risk of getting malaria multiple times, but also seemed to be at a higher risk of developing this specific cancer. This consistent co-occurrence was too strong to be a coincidence, strongly indicating that malaria infection, particularly chronic or repeated bouts, was somehow contributing to the cancer's development. Despite this long-standing observation and strong suspicion, the exact biological mechanism, the precise steps happening inside the body to link malaria to Burkitt lymphoma, remained a mystery for many decades. Scientists knew the what, malaria, especially Plasmodium falciparum, was linked to the cancer, but not the how. It was clear that the malaria infection was doing something to the children's immune systems, perhaps weakening them or overstimulating them in a way that made them more vulnerable. Pinpointing these precise biological changes became a critical area of research, as understanding the how could lead to better ways to prevent the cancer by tackling the malaria infection more effectively. Recently, some very clever scientists have made a significant breakthrough in understanding exactly how Plasmodium falciparum malaria can trigger Burkitt lymphoma. This new research, published in a respected scientific journal called the Journal of Immunology, has identified a key biological process. The work was led by Dr. Rosemary Rochford from the University of Colorado Anschutz School of Medicine. Her team compared blood samples from children in Kenya who had uncomplicated malaria, meaning they had symptoms like fever and chills but no severe organ damage yet, with samples from children who did not have malaria. This comparison allowed them to see what malaria was doing differently inside the children's bodies at a cellular level. The researchers focused on B cells, the type of immune cells that become cancerous in Burkitt lymphoma. They discovered something very important. The B cells of children infected with Plasmodium falciparum malaria had significantly increased levels of an enzyme called activation-induced cytidine deaminase, or AID for short. This enzyme, AID, actually has a normal and very important job in our immune system. When we get an infection, AID helps our B cells to fine-tune the antibodies they produce, making them better at fighting off that specific germ. It does this by making small changes or mutations in the antibody genes. This process is vital for a healthy immune response, allowing us to develop immunity. However, the problem arises when there is too much AID activity, or when it's active for too long, as seen in children with Plasmodium falciparum malaria. The constant stimulation of the immune system by the malaria parasite seems to keep these AID levels very high. When AID is overactive, it doesn't just make small, helpful changes to antibody genes. It can start making mistakes and causing damage to other parts of the B cell's DNA. One of the most dangerous mistakes it can make is to cause a break in a chromosome. Sometimes this broken piece of DNA from one chromosome can then wrongly attach itself to a different chromosome. This kind of genetic error is called a translocation. This is where the crucial link to cancer comes in. The research found that the high levels of AID in malaria-infected children make these B cells much more prone to a specific type of translocation involving a gene called MIC. The MIC gene is a very powerful gene that normally tells cells when to grow and divide. If the MIC gene gets broken off and moved to a new location, often next to a gene that is always switched on in B cells, it can become hyperactive. This means the MIC gene is constantly telling the B cell to grow and divide uncontrollably. This uncontrolled growth and division of B cells is precisely what leads to the development of Burkitt lymphoma. The malaria infection, by boosting AID, essentially creates the conditions for this cancer-causing genetic accident to happen. This new understanding of how Plasmodium falciparum malaria helps trigger Burkitt lymphoma is incredibly important because it points directly to a way to help prevent this cancer. If the malaria infection leads to increased AID, which in turn causes the MIC gene translocation and then cancer, then stopping the malaria infection in the first place could break this dangerous chain of events. This means that all the efforts we make to fight malaria, things like using insecticide-treated mosquito nets, spraying insecticides indoors, providing prompt anti-malarial treatment, and developing effective malaria vaccines, could also have the very welcome side effect of reducing the number of children who develop Burkitt lymphoma in these high-risk areas. The discovery provides powerful new scientific evidence to support and even intensify malaria control programs in regions where both diseases are common. Public health officials and communities now have an even stronger reason to prioritize malaria prevention. Imagine a child who is protected from malaria through a bed net or a future vaccine. Not only are they spared the fevers, chills, and potential deadliness of malaria itself, but their risk of developing this aggressive lymphoma could also be significantly lowered. This highlights how interconnected health issues can be, and how tackling one major disease can have far-reaching benefits for overall child health and well-being in these communities. It is important to remember that science is an ongoing process. While this discovery about AIDS and the MIC gene is a major step forward, researchers are continuing to investigate other ways that Plasmodium falciparum malaria might affect a child's immune system. Dr. Rochford's team, for example, is looking into further details of how these malaria-induced changes in immune function could promote cancer development. There might be other factors involved or other pathways that contribute to the risk. Continued research will help to paint an even clearer picture and could uncover further opportunities for prevention or treatment of Burkitt lymphoma and potentially other cancers linked to infections. In conclusion, the unraveling of the biological link between Plasmodium falciparum malaria and Burkitt lymphoma is a significant achievement with real-world implications. Understanding that increased AIDS activity driven by malaria can lead to the critical MIC gene translocation gives us a tangible target. It underscores the vital importance of malaria control and eradication efforts, not just for preventing malaria, but as a direct strategy for cancer prevention in some of the world's most vulnerable child populations. These findings bring new hope and empower health strategies in regions that have long been burdened by the double threat of these serious diseases, showing how scientific insight can lead to practical health improvements.